the exercises are based on, is a recent daily bill.
And if you go to search for this, just know that there is a UCSF Chimera software that is being replaced by UCSF ChimeraX. So there are two different software. ChimeraX is the much newer release that we'll be working with.
Yeah, you want to make sure that there's an X right here.
Phil can tell you all about it.
Want to talk about? Okay.
All right. Well, why don't we continue then?
The clothes. My book will be. Just pages and pages and pages. Take your towel and then I'll show you your clothes quickly. Yeah.
All right. So in this workshop today, we're going to be talking a little bit about computational structural biology, and mostly in the context of drug design, and how the program ChimeraX can be used in a lot of different activities related to that, and to learning about protein structures in general. First, just a bit of background about myself. So I'm Philip Cruz. I have a background at a university, in industry, and now I've been at the National Institute of Allergy and Infectious Diseases for the last 8 years, which is part of the National Institutes of Health in the United States. If you want to contact us, that information is shown on this slide. I've been part of the 3D visualization program here at NIAID for a while now, and Megan McCarthy is the lead of that program. And Megan, you want to say a little bit about yourself?
Sure. Hi, everyone. Happy to be here today. I am going to be supporting Phil. I am not a structural biologist by training. I'm actually a neuroscientist, worked in cell signaling, but I am in this world of 3D. I have learned, I'm learning a lot from Phil, and I'm actually probably going to learn a little bit today with all the new releases that have come out in the software that Phil's going to be demonstrating to you guys. I've been with NIAID back for just almost 10 years now, and I lead our program for 3D printing and biovisualization. So that includes 3D printing services. We make custom models of molecules and human anatomy that we will make for the investigators at NIAID. And then we have our biovisualization lab, where we do virtual reality, all kinds of new and advanced data visualization. There you go. So that's our BioVis lab on the left. We have a mobile app that we'll give a plug for at the end, Pathogen AR. And on the bottom right is Francis Collins, who is director of the National Institutes of Health, with a really nice model that we made of the SARS-CoV-2 variant last year that I'm supposed to have on my desk with me here, and I unfortunately do not. But that's sort of what we have products and services, and then we also develop different kinds of software applications. So in addition to the mobile app, we have a platform called the NIH 3D Print Exchange, where you can go and download files available for printing or other kinds of visualization. And then I'll just briefly mention our role in the African Centers of Excellence in Bioinformatics and Data Intensive Science. NIAID founded our site in Bamako in 2015, and we launched in Kampala, Uganda in 2019. And in Uganda, we had the opportunity to put in a big visualization lab, which I think there's a picture of here next. There we go. So that program is actually is dedicated to the program is dedicated to the hardware infrastructure and telelearning facilities for all kinds of remote training and instruction. The visualization lab was new, and that's to give people access to VR. And there's some really excellent work going on there, and we're starting up more of that in Mali. And I can see Grace from Macquarie University on, and she's done a lot with that. But what I'm going to leave that as a teaser for is towards the end of the application, at the end of the presentation today, Phil and I are going to be doing a VR demo. So I hope you will stick around. And whether or not you have virtual reality, it's an experience that you can have, at least to sort of share molecular visualization sessions remotely. But anyways, sorry, Phil.
No,
I'm
great.
giving away the good stuff yet.
One other thing I want to mention is that the ChimeraX is also good for looking at 3D medical images like CT scans. And we're going to also have a brief demo of this as well at the very end in virtual reality, where you'll be able to go inside of this lung and explore it from the inside out. And it's a very impressive experience. So here is the agenda for the workshop today. The first part is going to be lecture, where we'll introduce the ideas of cheminformatics and drug discovery. And then quick review of protein structure. We'll talk about the molecular file formats that are needed for storing essentially chemical information like a protein structure or a small molecule structure in a computer, because everything we're going to be doing is in a computer. And then at the end of the lecture, we'll be looking at 3 databases that contain protein structure information: the Protein Data Bank, the Uniprot database, and the AlphaFold database. And the reason I'm bringing those up and emphasizing them is all of them have information that's utilized by ChimeraX in analyzing protein structures. And then we'll start up ChimeraX, and this will be the demo part of the workshop. It's not going to be hands-on in the sense that I'll be able to review what everybody is doing at every step, but I'll try to go slowly through it so that those of you who have the program installed will be able to follow along. And it will cover just a fraction of all of the capabilities of ChimeraX, but it will give you a feel for a large fraction of the things that you need to get started with ChimeraX and feel comfortable working within it. And then, as Megan mentioned, we'll finish up with a virtual reality demonstration looking at some protein structures, both related to the exercise and in addition to those. And then finally, the medical imaging that I mentioned earlier. So at the top of the agenda, there's this word cheminformatics, and this is something that may not be familiar to you, but this is really kind of the essence of how computers are used in drug discovery. So here's a definition. And I'll just read it. Cheminformatics is the mixing of information resources to transform data into information into knowledge for the intended purpose of making better decisions faster in the area of drug discovery. So practically, what does that mean? It means it's a combination of chemistry, computer science, and information science. And it's important for the storing and searching of large databases. So large databases are important in the practical use of cheminformatics and essentially doing the practice of structural biology. And we want to find compounds, small molecules, and compare them for their similarity and diversity in order to help design new drugs. So why is this important? It turns out that cheminformatics methods have been really important for the creation of a variety of new important small molecules that have been developed into drugs. And they may also help by limiting the number of compounds that should be screened experimentally, because that's time consuming and expensive and just a lot of effort in order to screen large number of compounds through high throughput screening, which is HTS, or other approaches in order to find that one molecule that will eventually become a drug. So why is that important to simplify the problem? So if you happen to be working in a large drug company, they probably have between 10 to the 6th and 10 to the 7 compounds sitting on a shelf in their what's called their library. And 10 to the 6th, 10 to the 7th, that's 1 to 10 million. It sounds like a big number, but in reality, because the possible numbers of organic small molecules is so huge, the total number of possible drug-like molecules, which is called a chemistry space, is somewhere between 10 to the 60th and 10 to the 90th. So it should be obvious, 10 to the 6th, 10 to the 7th is a tiny, tiny fraction of the total chemistry space that you may want to look at to come up with a new molecule. And just to give you a feel for how big this number of 10 to the 60th to 10 to the 90th is, the total number of particles of matter in the entire universe is around 10 to the 80th power. So this is a huge, huge potential problem. So why would we use computers to help solve that problem? Well, the reason for that is to try to save time. Now, you might be wondering why there's such a huge range of 10 to the 60th through 10 to the 90th here. And one reason for that is there are multiple different definitions for what would be a drug-like molecule. One of those possible definitions is called Lipinsky's Rule of 5. And this is based on the work of Chris Lipinsky, who was a researcher at the pharmaceutical company Pfizer when he came up with this. And he looked at essentially the existing small molecule drugs that were in use and found some guiding limiting principles. So for example, there were less than 5 hydrogen bond donors in almost all of these, or less than 10 hydrogen bond acceptors. The molecular weight is less than 500 grams per mole, and the partition coefficient is, or called the log P, is less than 5. And let me briefly explain log P just for a moment. This is a measure of the difference in solubility of a molecule between water and some hydrophobic solvent. Usually it's octanol. And the reason this is important is because for a drug to be effective, it has to both be soluble in water so it can circulate in your blood, but it also needs to be somewhat soluble in hydrophobic material so it can transfer into your cells or arrive at the particular location that it needs to be effective. So a log P is just the log of the ratio between how soluble a compound is in octanol and how soluble it is in water. And so this is the last criteria in what's called the rule of 5. So again, this is one definition of what a drug-like molecule could be. And all of these parameters can be used to filter out any database you have of small molecules to leave things that are more likely to be drug-like. So now let's take a quick look at how drugs are created. And there's a variety of steps in this process, starting from target discovery. So this is trying to identify most likely a protein target that you're trying to affect with this new drug candidate. So now I mentioned that it's usually proteins. It can be other things also. It can be nucleic acids or even lipids, but most of the time it's going to be a protein of some kind. And then somehow you are trying to find small molecules that are hits which interact with or affect somehow the target. And at this stage, you're typically screening huge databases of compounds. So from all of the hits, which are kind of the ones with the best properties, you want to narrow that down to what are called leads, which is a much smaller number, and then optimize the leads. And at this point, you're finally ready to go into the clinic. So there's a preclinical phase, and then phase 1 drug trials. And phase 1 is usually a relatively small number of participants, human participants in the trial. And this is mostly to test whether this new molecule is toxic or not, to see if there are adverse effects. If the drug candidate survives that, you go into phase 2, which is a much larger group. And now you're really looking to see whether the molecule is effective in treating the disease that you're targeting. If you survive that, then you go into phase 3, which is a much larger and more rigorous phase. And finally, if all of that works out, then you can go through approval and then launch the marketing and selling of a new drug. So this is the way it's done in the United States, but at other places around the world, it's a very similar set of steps. Oops, wrong way. So the first several steps up through lead optimization, the thing that's most important usually is looking at the drug target, the protein. And then starting from the preclinical stage, the most important thing that's being concentrated on is the actual drug candidate itself, the small molecule that you hope will become a drug. And these two different phases are referred to as the discovery phase and the development phase. And computational drug design, which is what we're going to be talking about today, is related through all of the phases through the preclinical phase. And within computational drug design, there are various sub-specialties. Early on, what's most important is modeling. And the hands-on exercises today, we're going to be doing a lot of modeling of protein structures. The next step is screening, where you're trying to find those hits and leads. And we're also going to be talking today some about how to analyze screening results. And then finally, the latter steps are refining the leads through optimization and doing further analysis. So again, these are the steps that are done using a computer and programs like ChimeraX. Now, computers are also used in the later development stages, but that's a process for a different workshop. So I hinted at this earlier, but you can think of this process as like a funnel that starts out very big at the beginning, where you can have millions of compounds that you're considering. And as you go through the funnel, through the process, there are hundreds of compounds. And then maybe 5 or fewer compounds in the later stages. And then hopefully at the end, you get to that single approved drug. Now, so it's really important to know that computers are important in quickly getting between, one moment here, I lost my cursor, there it is. So computers are important in quickly getting through these early stages, screening and getting through. And it's really important to do that, not only for time, but also for cost, because this reverse funnel, which starts out small and gets big as you go along, means that the cost of the drug creation process goes up very quickly with time. And most drug programs end in failure. So if you're going to fail, you want to fail as early as possible. And the methods we're going to be talking about today not only help you get to the end quicker, but it also helps you fail faster if it's unlikely that that particular drug program is going to be successful. So here's a variety of drug design methods that are used with computers. And these are all in the earlier stages like we were talking about on the last slide. And there are two broad categories of them. Structure-based, and structure-based means that you have a protein structure or a target structure available for you to study. And then ligand-based, which means that you don't have a protein structure available. But what you do have is activities measured in a laboratory against the target that you're interested in. So probably the best known of the structure-based methods is docking. And this is where you take a library or a database of small molecules that are drug-like molecules, and you try to dock them into a binding site or a pocket on a protein structure. And it's based on identifying an active site and analyzing the active site features. And there are also other structure-based methods, which I'm not going to mention today. And I'm also not going to say much more about the ligand-based approaches, because this is a topic for another day, and it's not really where ChimeraX has its strength. But I am mentioning these kind of because they are extremely important. It turns out that the ligand-based methods in practice are typically even more powerful than the structure-based methods. And I know that's somewhat counterintuitive because you don't even have a protein structure. But the difference is with the ligand-based methods, you do have experimental data from the laboratory that helps guide these approaches. So that's what makes these very popular. But again, that's a topic for another day. All right, so I know many of you don't think about protein structures all the time. And so I just wanted to have a quick review of protein structure. And there's another reason I'm showing this slide, because the first exercise in a few minutes, we are going to be creating this image using ChimeraX and show you the steps in order to do that. So, you'll all know that a protein structure is a chain of amino acids. In this case, there's 3 alanines, alanine 1, alanine 2, and alanine 3. They're numbered from the N terminus, which has an ammonium group, and down to the third one, or the end of the chain, which is called the C terminus, which has a carboxylate group. And the N terminus has a plus charge, unless it's been modified. Similarly, the C terminus has a minus charge. unless it's been modified. And connecting amino acids are what are called peptide bonds. And another name for these is amide bonds. And so those are shown here, the connections between adjacent amino acids. All right. And we refer to the backbone, which defines the path that the overall protein takes through three-dimensional space, as the backbone. And the backbone atoms here are colored in red. And you'll notice you can also simplify that just by looking at the positions of the alpha carbons on each atom. One of them is labeled here, but there's one on each amino acid, here, here, and here. And at this point, I'd like to mention one other definition. I'm going to use the words amino acid and residues interchangeably today. Residues refer to single monomers in biopolymer chains. So for proteins, a residue is an amino acid. For nucleic acids, a residue is a single nucleotide. And so again, each amino acid consists of an amide. There's an alpha carbon, and then coming off of the alpha carbon is a side chain. And the next slide is just highlighting the side chains in green. Now for alanine, the side chain is just a simple methyl group, a carbon with 3 hydrogens attached. Okay. So this is the very basics of protein structure. And we want to be able to show protein structures in a computer. And if we're doing drug design, we also want to be able to do look at small molecules, the drug candidates in a computer. So how do we do that? And that means that we have to put this information in files somehow. So I'm just going to cover in the next several minutes the common structure file formats. So these are chemical structure file formats. And sometimes you'll see them referred to as connection tables, because not only does it show where the atoms are, it also indicates where the bonds are, which are the connections between atoms. And again, we're going to go through each of these for a few minutes. I know this is kind of a boring topic, but it's very useful to understand some of these concepts when you're actually working with structures, chemical structures of any kind on a computer. So the first one of these is the PDB, or Protein Data Bank format. And this is probably the most important one for use with proteins. And it's the one we're going to be looking at the most today. While it can be used with ligands, it's not really the best one to use if you only have ligands. And here's where we're going to have another definition. A ligand is a small molecule which interacts with a larger molecule, such as a protein. So I'm going to use the words ligands and small molecule or drug interchangeably today as well. So again, when you see the word ligands or hear the word ligands, that's referring to the small molecules. Another choice is to use when proteins and ligands are together. So you have a structure which has the protein and the small molecule bound to it. And the last column here indicates whether there's three-dimensional information in this file format. And the answer is yes. For PDB, it does have three-dimensional information. And most of what we're going to be looking at today is three-dimensional. Okay. The next format is MOL2. It's not as widely used as PDB, but it does have some advantages. One of those is that unlike PDB, it is good when it's used with both proteins by themselves and ligands by themselves, but it does check all of the boxes here. The next file format is the SD file, or a structural data file. This is the most used format for having small molecules or ligands. And most of the databases of small molecules allow you to download their contents in this format. And again, this one also has three-dimensional information. And then the last format is called SMILES. And it's only useful with ligands, and you'll notice it does not have three-dimensional information in it. So what are the essential types of data in a molecule file? Well, we need to know something about the atoms, things like what element it is, what the name of the atom is, and its X, Y, and Z coordinates, where it's located in space. We need to know something about the bonds, which are the connections between the atoms. And with proteins and nucleic acids, we need to know something about the residues. What's the type of residue? What's the residue number? What's the chain name of the protein chain? So these are the kinds of important information that have to be in a file. So let's take a look at the PDB file. The PDB file comes from the protein data bank initially. And so that's a database of protein and nucleic acid structures. And we'll take a close look at that in a few moments. But these files normally have a 4-character identifier, although that's going to be changing soon. But let's just look at a small excerpt from one of these files. So the lines that come from the word, that start from the word atom, just as you might expect, have information about an individual atom. And they are numbered sequentially from 1 through n, which is the total number of atoms in the molecule. And you'll see the ... here. That means that there's lots of them. There's going to be hundreds or thousands of lines start with the word atom. The next column here is the name of the atom. In protein structures, each particular atom has a specific name. The next column is what type of residue or amino acid this atom is in. In this case, it's an isoleucine. And then J indicates the chain name. In this case, this is coming from chain J. And then there's 3 more important pieces of information here. There's this 5.8, 123.9, and 147.4. These are the X, the Y, and the Z coordinates of this particular atom in space. So if we go back to the previous slide, essentially we have all of the information that's listed here except the bonds, which is the connection between atoms. And it turns out that for proteins, that information doesn't have to be included in a PDB file because we know what an isoleucine amino acid looks like. So we know just from the basic structure of isoleucine which atoms are connected. So it's not important necessarily for that information to be in the file. However, for small molecules, for the drugs or the ligands, we don't know what atoms are connected. So those are given by lines that start with het ATM or het atom. The rest of this information is the same. The residue name here, HOH, is telling me that this is a water molecule. But since we don't know what atoms are connected in some arbitrary molecule, the het atoms also have lines that start with connect, and this tells us where all the bonds are. Okay, so that's the PDB format. Now we'll take a quick look at MOL2. It has similar information, but it splits the information out into atoms, bonds, and substructures, which are residues. So it has similar information. 1, 2, 3, a count of the numbers of atoms, the atom name, and the XYZ coordinate. And it says which residue, ASN1, this is associated with. And then here is an explicit listing of the bonds. But oops, unlike the PDB format, all bonds in the molecule are listed. And then we have the substructure or the residue information. And it's saying that the sequence of this molecule starts from residue number 1, which is ASN, and then continues down through the rest of the structure. So briefly, that's what MOL2 files have. So now let's look at this SD format. Again, remember, this is just for small molecules. And the first part here, GDP, this corresponds to the molecule's name. And you can ignore this line. This is just telling basic information about the molecule, like how many atoms or how many bonds are in it. But we see similar information like we saw in the previous formats. Here's an X, a Y, and a Z coordinate, and here's an element type. And then this next section down here lists the bonds, okay? One additional thing that the SD file has is it can have arbitrary data fields in it. So this can be any kind of information. It could be text, it could be numeric. So for example, here, there's some sort of energy associated with this particular molecule, and here is its value. So you can have name value pairs included in an SD file. Now here's the last format of the 4 that we're going to look at, the SMILE strings, which is an acronym. You can see what it means here. It was designed by David Weininger in the 1980s, and it's referred to as a line notation. Again, it's only for small molecules. And it's just a system using simple ASCII characters to describe what a molecule looks like. And it turns out that most molecule editors and most molecular modeling programs can read and convert back into 2-dimensional drawings or 3-dimensional models of these structures. But there's only 2-dimensional information contained within. So here is an example. Here is the SMILES string for this particular molecule. And as you can see here, it looks a lot like a chemical formula. Again, it's 2D only, and we don't use this for proteins because there's no information about residues, about amino acids in a SMILES file. So let's take a look at some online web sketchers that understand SMILES. There's 3 of them here that I'm listing. There's lots of other ones. And you can see their URLs. And we're going to do just a little quick test here. So I'm going to stop the presentation for just a moment. And I'm just going to do a quick selection of this SMILE string. And here is the first one of those web sketchers, which is the PubChem sketcher. And I'm just going to paste this string into here. And this sketcher knows what this structure is just based on that string. And a lot of these sketchers will be able to convert this two-dimensional sketch. You can see here it's just very flat. Oh, I messed it up, sorry. But it's just flat, but you can export it to three-dimensional structures from many of these sketchers. So almost all programs or online web services understand SMILES for small molecules. So it's a very important structural format. If you play with it a lot of people are expert at just seeing a molecule and can just write it out because on their own, because it's just so much like a chemical formula that you may have seen in your beginning chemistry classes. Okay. Let me see. Okay, so I've been talking about small molecules some and how they would be used in trying to identify new drugs. So where do you find small molecules? You don't want to have to sketch each one of them individually or come up with SMILE strings and put them in a web sketcher. So here are 3 databases of small molecules, and they're searchable. You can filter on them. The first one, probably the most important one, is the zinc database. It has over 230 million compounds within it. They're ready to use for drug design campaigns. They have 3D formats, and most importantly, they're purchasable. That means that these are essentially a combination of catalogs from many, many different companies that sell small molecules. PubChem is another database. It has nearly 100 million compounds. A lot of these don't overlap with zinc, and they may be hard to obtain sometimes, but this is another great source. And finally, there's the Chemical database. This is the chemistry database from the European Molecular Biology Laboratory. It has nearly 2 million molecules, but the advantage of this database is that each one is known to be biologically active with some drug-like properties. So that means that there are laboratory measurements against specific protein targets, and you can search this database by target. So if you are trying, if your target in your drug design or your drug discovery, if the target you're working with is similar to some other target that's already been studied, this could be a good place to start with to try to get molecules to begin with and then go from there. All right, so now that we've been talking about databases, let's move on to something that's more relevant to what we're going to do today. And that's 3 free databases that contain protein structures. And all 3 of these are very important. They're critical for obtaining the structures that we're going to be using. And I'm going to spend, again, some time going through each of these. And later on in the hands-on exercises, we will be looking at how ChimeraX utilizes the information in each of these. So the first of these, and probably the most important for our purposes today, is the protein data bank. It has experimental structures of proteins and nucleic acids. And it's important to mention that these are experimental structures. These are not computer models. And then the next database is Uniprot. And Uniprot, they try to be a comprehensive resource of sequence and function for proteins. And this is a huge database. It's partially curated, and it has lots of additional information besides any structural information. Lastly, there is the AlphaFold protein structure database. This is very new. This one just became available about a month and a half ago. But this is really groundbreaking in its ability to help with structural biology. And it has, this comes from a computer model, but it has predictions for the entire proteomes of 21 key organisms, including humans. So let's look at each of those in turn. First of all, the term PDFB refers to two different things. As we saw just a few moments ago, it refers to a file format that can be used to represent the structures on the PDB website. The original type that we took a closer look at has the extension .pdb, but there are two newer ones. There's the .cif and the .xml, but PDB will soon be retiring this particular format, replacing it with the .cif, but the information and the ideas are very similar to what's already in the protein data bank. So as I mentioned before, all of the structures are experimental, and currently they're all represented by a 4-character accession code. Although again, this will also be changing soon to go to a longer identifier, because they're running out of 4-character codes. So the protein data bank is managed by a group called the Worldwide PDB Organization, and that's abbreviated WWPDB. And there are several local mirror sites for doing the searching and downloads. And this particular logo I pulled off of their site because the protein data bank just turned 50 years old. And so they have been collecting protein structures for a very long time now. And that's why they need to modernize and update both the file format and the number of characters in the identifier for each structure. Now, in the United States, the RCSB is the organization that hosts the PDB. Excuse me. In Europe, it's the protein data bank in Europe, and in Japan, it's the protein data bank in Japan. And these are the URLs for each of those, and just try each one to see which one seems to have the best performance for you. But the information contained within them is the same, but the interfaces and the way the various pages look will be somewhat different. So let's take a look at one of these. And so I'm going to look at the RCSB homepage. And frankly, this is because this is the one that I'm most familiar with. And there's a lot of overview information on here. And the bottom left is some of the most recent additions into the database. up here at the top, they highlight a structure, a molecule of the month. And this is curated by David Goodsell, who does fantastic drawings of protein structures and does wonderful explanations of the various aspects of protein structure and function. But what's probably the most important on this page is the search field, where you can try to search for a structure that you're interested in. So I did a structure for 4DPD. So this is the 4-character identification code or accession code for a PDB structure. And just for a hint for what we're going to be doing later, this is a molecule that we're going to be looking at a lot today in the hands-on exercises. So there's a variety of different information in this page. You can get information, you can view the structure here. You can download the files here. And here's information on the literature reference, which refers to this structure. And if you scroll down on this page, at the bottom of the page, there's additional information, like information about the protein that's contained within, information about any small molecules which are in this particular structure. Here's some sequence information. And one other thing of note here is a link to the Uniprot database, which has information relating to this particular structure. So if we click on this link, let's take a look at Uniprot for a moment. So I've repeated the mission statement of Uniprot here. Uniprot really tries to be comprehensive, a resource of different protein structures and the functional information that's known about them. So there's a little over 500,000 manually annotated structures in the database, but they also do automated literature searches to add them. And there's an additional over 200 million structures with entries in the Uniprot database. So this can be, again, a very comprehensive listing. So let's take a look at the Uniprot entry for the protein structure that we were just looking at. So here is the page, and I'm not showing the scroll bars here, but this page will scroll down to give you lots of additional information. But one way of quickly navigating through it is you can click on the feature viewer here. And one of the things, information in the feature viewer is the structural information. So this will list the protein structure, and this is a 3-dimensional viewer, so you can move this around with your mouse. This indicates that there are multiple different PDB structures associated with it. And in the sequence viewer in it, up here at the top, you can highlight individual residues with the mouse and get feedback. And you can also click on a particular residue in the structure and get feedback on the sequence. Okay. Now I'm going to look at one. Oh, let's see. Yeah. So I'm going to look at the feature viewer for a different protein here. This particular protein has a section for variants in the feature viewer. So we've already looked at the sequence viewer, but the variant viewer has a listing of a variety of different variants from the literature and showing where in the sequence the different mutations are for these variants. And you can click on these and see them in the sequence. So this is a very powerful tool to see and to identify and to learn more about what variants of your interesting structure have already been published. Okay, we're going to go back. We're going to look at one other thing here in this particular Uniprot entry. I'm going back into the sequence viewer again. So whenever available, Uniprot also has links, like I mentioned before, to structures in the PDB. But even though the AlphaFold database is very new, it already has links to AlphaFold structures if they're available for this particular protein. So we can click this link here, and now we'll take a look at AlphaFold. So a little background information. AlphaFold is based on the AlphaFold database is based on the program called AlphaFold II. And it's an artificial intelligence, it's a deep learning program that does protein folding based only on sequence. And it was developed by Google's DeepMind subsidiary. Now, every 2 years, in the area of protein structure determination, computer protein structure determination, protein folding, there is a competition called the CASP competition. And the most recent one of these was CASP 14 in 2020. And AlphaFold II predicted over 2/3 of the proteins with greater than 90% accuracy in the competition. And this journal reference down here in Nature, which was just published back in July, explains more about the method that was used. Now, what do I mean by a competition? The way this works is there are experimentalists who determine protein structures in the laboratory and then submit them to the competition prior to them getting published in the PDB. So these are protein structures where the structure is known, but it hasn't been published yet. And then dozens of different groups try to predict these structures using their programs. And AlphaFold II did much better than all of the others, did much better than any others have ever done in this competition. So actually you hear words like remarkable when discussing this idea that it was able to predict over 2/3 of them with greater than 90% accuracy. So AlphaFold II is really a game changer in the area of drug design and structural biology. So, and it's brand new. So we're going to be seeing in the next few years how the various ways that it's going to be used. But the European Molecular Biology Laboratory and the European Bioinformatics Institute and DeepMind collaborated to make AlphaFold II structure predictions for the complete proteomes of humans and 20 other species, like I mentioned earlier. And so they have a searchable database, and you can access it again here. And this database was described in the same issue of Nature with this reference. So here's the home page, and there's a lot of great information here. This website was really well created. There's a lot of really useful information in the FAQs. Unlike many databases, which often don't have a lot of useful information in the FAQs. I would really encourage any of you, if you're using AlphaFold, to look through those, because there's really great guidance on how to use it in there. Here's a list of species included in the AlphaFold database. As I mentioned before, there's Homo sapiens, but there's also Plasmodium falciparum, which is the basis for the exercises that we're going to be doing a little bit later. And in fact, the protein we're going to be looking at later is is dihydrofolate reductase thymidylate synthase. And here is the AlphaFold database entry for that particular protein. And you can see here, you can download a PDB format file containing the structure. You can look at the alignment that it's predicted from. You can also download the newer MMSIF file. There's a variety of information here. And there is also a 3D structure of the protein that was predicted. And so you can use a mouse to manipulate and move around, look from different angles at this structure. And it's shown in what's called a ribbon representation or a cartoon representation, where just the path that the backbone of the protein takes. And you can see some of it is very compact. You can see some beta sheets down at the bottom and alpha helices throughout it. The parts that we would normally think of as a globular protein. And some parts are loops, which kind of extend off into space. So the color of the different parts of the backbone are coded according to how confident the model is in its predictions for that part of the structure. So you can see the colors range from blue down to orange, which is below 50%. So there's very high confidence in the blue parts. And in fact, if you compare this with the crystal structure, it's very close, very close. But if we look at the low parts, the orange parts, well, that includes this loop that sticks off into space. So when you're looking at that part of the structure, don't put really any confidence in how that goes in 3 dimensions. And in fact, there is a good chance that in vivo, that part of the structure is disordered. It's just moving all over the place, unless this protein is interacting with some other protein, which may lock those loops into place. And I want to mention the limitations of AlphaFold as it currently stands. And the first one of these is probably the most important one. And that is that AlphaFold has not been validated for predicting the effects of mutations, especially of things like SNPs. So if there is a structure that you're interested in, or if there's a protein that you're interested in for which there's already a structure, don't expect to be able to just mutate one amino acid and then have AlphaFold calculate it, because it's not designed to do that. And it's probably less useful to do that than to just mutate the amino acid in ChimeraX or some other modeling program. The other thing, we'll go through these quickly. I mean, AlphaFold only handles single chains at a time. And there are regions that probably are disordered or unstructured in proteins in isolation. And as we saw in the previous slide, AlphaFold will give low confidence predictions for those. Some proteins have more than one conformation, but AlphaFold will only produce one of them. And AlphaFold will not predict the positions of any non-protein components, such as cofactors, metals, ligands, solvent, DNA or RNA, or any post-translational modifications. Okay. Now, before we move on, I do want to take a little side journey and talk about what to do for computer models before AlphaFold. And that's the idea of homology modeling. Now, homology modeling is where you use an amino acid sequence and a database of experimental protein structures to construct 3-dimensional models. And that's done based on the sequence homology of your unknown sequence against proteins of known structure. And there's many, many online servers that do this. And I just want to mention there are other types of protein modeling, such as secondary structure prediction for local structure only. Again, there's lots of different servers on them. You'll find them with a Google search. And then there's also ab initio protein folding, which is really computational difficult. And I'm going to give a very high level concept of homology modeling on the next slide and give an example from one website, because I don't think these are totally obsolete with AlphaFold. And I just want everybody to have some exposure to them. So the idea is you have a database with templates and you're trying to find an unknown structure, but you do have a sequence. So your unknown sequence is aligned to the known sequences, the sequences of the known structures in the database. And based on that alignment and structural considerations, a structural model is created. Typically, multiple templates are used, not just one, to give better confidence. And then the backbone is refined so that there's not anything that's sterically or otherwise unusual. And then as a last step, side chains are added by these programs. One of the best, if not the best, the one that I use the most is called iTasser. And the link to it is shown here. Essentially, all you have to do is go to this web page, register for a free account, and then paste your sequence into this field here and click go. And it takes usually 2 or 3 days, but it will generate a series of models that you can take a look at. And so this is still a good way of generating sequences of structures from sequences that you don't have already in one of the databases. And there's some good rules of thumb or general guidance for how good the model quality you can expect from sequence homology methods. If the sequence homology is greater than 30%, the confidence is usually good to great from these programs. Even down to 25 or even 20, the confidence is low, but it may give you a general idea of what the three-dimensional structure is. And below 25, probably the confidence is very low. So be very careful in using the structures that are produced. And once you have a structure, you want to consider this. You want to look at if there's any steric or electrostatic clashes in the model. You probably have some bench data and you want to see if there's anything that contradicts that in the model, or just general protein structure information itself. And then the various programs will give you their own scoring, and you can use that also to help you guide to determine how much you should believe the model that's created. But let's say you do have a model that's that you have reasonable confidence in. So you'll always ask the question, is my model good enough? And it turns out that it depends on what you're trying to do with a model. So are you trying to do drug design? Well, in order to do that, you need a fairly accurate representation of a binding pocket or a binding site. And the answer is, if the sequence identity is pretty high, like greater than 50%, yeah, usually it can be. Now, if the sequence ID is lower, but it's accurate around the site of interest only, then it may also be good enough. If you have reasonable confidence but low sequence idea, you can still use it for a variety of tasks, sometimes helping identify what the function might be, but it's probably not going to be good for your drug design project. And just like with AlphaFold, the structural effects of point mutations will not be modeled accuracy by homology modeling programs. And again, you need to use something else like a force field method, but that's beyond the scope of today's talk. Okay, we're getting close to the end of this section, but I want to talk about some free protein modeling programs. There's ChimeraX, which we're going to be using today. There's also one called VMD, which is useful for studying molecular dynamics, the force field methods that I talked about in the last slide. And there's another one called PyMol, which many of you probably have used already, which is also very popular. And here are the links to those. But again, we're going to look at ChimeraX. And I think we can stop there and take a 10-minute break at this point. So were there any questions in the chat? I haven't been monitoring that.
No phone. I don't see any questions.
Okay, great.
So a
There's
question,
some now.
Okay.
Bill, a question just came in now. Is ITAS used for protein sequence or nucleic acid sequence?
Protein only. Okay, so let's take a quick break, and about 25 after the hour.
Please, I have a question, sorry.
Oh, go, sure, go ahead.
Yeah, thank you very much. So in the last slide.
Excuse me, can you speak up? You're very quiet on my side.
Okay, it's Ayub Ksuri from Institute Pasteur of Tunis. So in the last slide, you said that the quality of you judge the quality by percentage. For example, under 50% it's good, under 20% it's low, but you don't see that it's depending on the length of the sequence here. Yes, in this slide.
Yeah, so what is the question then,
Yeah,
based on this?
You don't see that the quality is depending on the length of the sequence, not only on the percentage of sequence and data.
You're absolutely correct. This is an oversimplification, and not only does it depend on the length, but sometimes the protein, your sequence only covers part of the protein structure. Sometimes, which is much more common, your sequence is much longer than the part of the structure that's in the structural database. And so these programs often have difficulties with that. Sometimes if you have a long sequence, some parts of it will match one set of templates and another part of it will match another set of templates from completely different proteins.
Yes, so I cannot consider this table as general rule.
Right. This is just a simple rule of thumb or a simple guideline, but yes, you're absolutely correct. There's much more to it.
Okay, thank you. Thank you very much.
Great. Okay, well, I'll see you back in a few moments.
Okay.
Okay, so let's continue. For any of you that may have joined later, here is a link to download the ChimeraX program. And we want, when you get to that page, you want the daily build. And if you download it, you can follow along with the steps here. Now, this is not going to be hands-on in the sense that I'm going to be able to stop and help people because there's too many. But I'm going to try to go slowly through the various steps so that you can keep up as we go through it. Now, just a brief overview of ChimeraX. As you'll see when you try to download it, there are Windows, Mac, and Linux versions. It has both a graphical interface and a command line, and we'll be using both of those heavily. It's also very commonly used for fitting cryo-EM density maps to protein structures. And as we'll see at the end, and this is very exciting, is that you can go into your structures in virtual reality and also in medical, and also you can view medical images like CT scans in ChimeraX and also go into those in virtual reality. So here's the first screen in exercise 1, where we're going to review the protein structure. We're going to create the scenes that we looked at the very beginning when we reviewed protein structures. And in the presentation, the ChimeraX window is typically going to be on the left, and then the instructions for what you're going to do are going to be on the right. So I'm going to end the presentation now, and we're going to do this in ChimeraX. Oops. Okay, great. So, as you can see, the first step is we're going to build an ALA peptide. So if we go up to the top, I'm going to make this a little bit bigger. If we go up to the top to the tools menu, we go down to structure editing, build structure. And it gives this little, it puts this window onto the screen for you. And I want to click to add a peptide. So in the box where it says protein sequence, type AAA, and then click apply. So now it's going to ask, how do we want to set the phi and the psi angles between the amino acids? So I'm going to shift click all 3 rows in this table. And then I'm going to go down here and it's where it says seed above phi and psi residues. I want to choose anti-parallel beta sheet. And then if and that tells me what the angles are. And if I click set, that matches now what you should see in the presentation for what the angles should be. So then, let's see. So then just click OK. And there it is. So in this case, they were all set to the same. And this doesn't quite match yet what we have in the view from the earlier in the presentation. So first of all, let's do some things to affect the color. So I'm going to go with Actions, Color, and go to All Options. And in this dialog box, I want to click Background and make it white. And now we have a white background. And I'm going to go back to coloring atoms and bonds because that's more useful. And then we're going to close this. Now I'm going to give a first example of using the command line. So the command line is at the bottom of the screen. And I want to add hydrogens. And the command for that is ADDH. So if you type that at the bottom and hit enter, you now have hydrogens added. So if you go on to the next slide in the presentation, there's a review of how the various buttons on your mouse work. So the left mouse button does rotation around the X or Y axis if you move it. But one thing that's really powerful and that it's unique to Chimera and ChimeraX is if you put the cursor in one of the corners of the graphics window and move the cursor, now it rotates around the Z axis, which is the axis coming out of the screen. So you can easily, without having to do anything fancy, move around Z or around X and Y. The middle mouse button is Translate, which allows you to move the molecule around on the screen. And the mouse wheel is the Resize button. And I probably should mention at this point, working with virtually any molecular modeling program, it's really important to have a 3-button wheel mouse. It's possible to do this on a touchpad or other kinds of mice, but every molecular modeling program I work with is designed for a 3-button wheel mouse, and it's just a much better way of doing things. The last thing I want to mention is if you point to an atom and you control click, so can hold control and click on it, that selects that atom. And you can see that there's a light green highlight around the atom. And if you want to clear any selection, you just control left click anywhere in the background. So just put the cursor anywhere in the background, control click, and that removes the selection. And there's one other very powerful tool within ChimeraX related to selection. So I'm going to click this atom again to select it. And the up and down arrows on your keyboard will extend or contract the selection. So if I hit the up arrow once, it selects that entire amino acid. And if I hit the up arrow twice, it selects the entire protein chain. So that's a really valuable tool for selecting chains and just individual amino acids using the up and down arrow based on a single atom selection. And the down arrow goes back the other way to a single residue or the original atom that you selected. But I'm going to click again in the backdrop to clear the selection. Okay, so what I want to do is rotate this so it matched pretty much the original view that was in the presentation earlier. Okay, that's pretty close. Now let's color the different residues. So I'm going to click on the leftmost alpha carbon to select it. So that's Control click. And then I'm going to press the up arrow to select that residue. And then I'm going to go up to the Actions menu, color yellow. Then I'm going to go to the middle mouse button, repeat. Click on it, the alpha carbon, up arrow, Actions, color cyan. And then finally, the third one, click on the alpha carbon, up arrow, Actions, color magenta. And now I want to clear the selection. So now we have the residues all colored individual colors, but it still doesn't quite match the original scene. So I go to Actions, color by heteroatom. And heteroatom is a term used in structural biology to refer to anything that's not carbon. So that's a useful shorthand sometimes. And then this gives us the molecule representation that we saw earlier in the presentation. Pretty easy, right? And now, but this is a three-dimensional representation here that we just created, as you can see by moving it around. So the final step is to add labels. So we're going to do this from the command line. And on the next slide, it tells you what command to type. So it's label number 1. And number 1 refers to what we see here is the model ID number. So ID 1. And we're going to label, we're going to put on residue label. So I'm going to type residues, color, line, height 0.4. Okay. And when I type enter, you'll see that there are now labels. And then this view very closely matches that original view that we had earlier in the presentation. Okay. So one final thing I don't really like where the second alanine was, or the first one, because they're in front of the structure. So let me just move them. So if we go up to the top, you'll see that there's a series of tabs above the icons. And I want to click on right mouse. And this allows me to assign different functions to the right mouse button on the mouse. So in this case, we're going to look interesting. So I did this, I prepared this using a Mac. So it appears that the move label icon isn't on the Windows version. I'm going to, let's see, maybe there's more to the right here. No.
Bill, did you say the move label icon?
Yeah, am I missing it?
Yeah, it's under annotation, distance label, move label.
Oh yeah, it looks different here. That's right, I was looking for the green color. Oh, there it is. Okay, so if I click the right, sorry, thanks, Megan. Left click on this to select it. And now if you use the right mouse button on a label, you can move it around. Okay, and I wanted to show this here because we're going to be using the right mouse buttons later on, mainly in the virtual reality demonstration. Thanks, Megan. Finally, we're going to color the backbone red and the side chains green. So, and now we're going to use some menu commands. So far we've used icons, we've used the command line. Let's use some menu commands, more menu commands. So the select menu is a very powerful feature in Chimera to precisely control what you're selecting. So I'm going to go to Select, Structure, Backbone. So now just the backbone is selected, and then I can Actions, color red to make it red. And we could do the same thing with the side chains. Select, Structure, Side chain only, and now we're going to color those green. All right. And as a exercise for you, see on your own sometime if you can figure out how to get the backbone colors back and only color the side chain green. All right. So we have this the way we want it. So now I want to save an image file for putting in my PowerPoint presentation. So I can go to File, Save, and in Files of Type, there's lots of different choices here. And notice there's a scroll bar here on the right. So there's a lot of different files. You can choose PNG image, and it will give me some additional options here so that I can adjust the size of the image that gets saved, whether I want to preserve the aspect ratio so that it doesn't change its shape. And the super sample indicates how accurately I want to indicate the structure. You'll get a bigger file if you choose 4 times, but you'll get a better presentation of the structure. And you can notice you can also turn on a transparent background here. So you can navigate to where you want, you can give it a file name and click Save. And the options that appear at the bottom will depend on what file type you choose. I'm not going to do this because I already have this file, but if you want to, please go ahead. Okay, so that's the very basics. Now let's get something more interesting. We're going to start working with real protein structures now. And the exercises that we're going to do today were inspired by this particular poster from the recent meeting. And it's looking at analysis of the Plasmodium falciparum DHFR gene. There are known structures for that gene, and there are 4 specific mutations that were looked at in this study. So we're going to take a look at this structure in ChimeraX and see what we can find out about it. So if we go to the next one, again, I'm not going to show the presentation on my screen anymore. Hopefully you're all following along. This is from a PDB entry called 4DPD that we looked at earlier. And what we're going to do now is we're going to load that into ChimeraX. So what I want to do is I think I want to close this and start from a fresh ChimeraX session. And down here on the command line, I'm going to type in open and then the PDB code for DPD. And there it is. And there's several things that I want to say about the screen as it looks right now. If you go to the next slide in the presentation. So by default, the protein backbone is shown as a ribbon. And any ligands or small molecules are shown as sticks. Now, if there are ligands in this structure, that usually means that that area is kind of important for the structure. It might be a binding site. It may be where a cofactor binds. So any of the protein side chains near the ligands are shown as sticks, but most of the protein side chains are not shown. So this is usually a very common and useful way of representing the structure. You can zoom in on it here. You'll notice that there are some little red balls or red dots. Those are the water molecules that were in this structure. And really very important, if we hover the mouse cursor over a part of the structure, we get a little pop-up menu that tells us what this is. So this particular amino acid is isoleucine 142 from chain A. So that's what the /A at the beginning means, is chain A. Okay, so this is kind of the basics. You'll notice here at the bottom is what's called the model panel. And it shows us the name of what we just loaded, its ID. We can change the color here by clicking on this little box. The eye indicates whether it's visible or not. And so we can turn it on and off, visibility. And the little pointer icon column is whether it's selected or not. So we can select everything in that structure very easily with that icon. Moving up, the top part is the log. And the log contains a lot of information about the structure we just entered. And plus it's showing any of the commands that we've done so far. So it knows that it's loaded two different chains, chains A and B of this protein. But look, there's also a link to Uniprot here within it. It also lists what non-standard residues are in here. So this is going to be things like, these are going to be the various ligands. And so you can get additional information about these very quickly. So going on to the next one in the presentation. The way you work with ChimeraX, and there's really kind of a common paradigm of how to use it, is first you select what you're interested in acting upon, and then you do an action. And those actions only happen to the selection. Now, most actions will act on everything if nothing is selected. And I'm on slide 72 now, if you're following along. The select can be either from the select menu that we looked at a few moments ago, where there's lots of different ways of selecting things. You could select on the screen, as we saw earlier. We can put the cursor somewhere and control click, and that highlights that to select it. Or you can use commands to select with a specialized syntax, but I'm not going to really get into that again. Now, the actions you get to from either the actions menu or from the command line. So and we're going to go through several of those now. So if we look at this structure, turns out that there's two chains here. It turns out there's two domains. It's really a lot more complicated than we're really interested in probably. So what we're going to do is we're going to use various selections and then actions to simplify the structure. So the first thing we want to do is select chain B. Now we could do this from the select menu. Select chain B. Okay, I'm going to clear that selection by control clicking in the background. Or I can click this B here in the log, and that will select. Or finally, I can do it from the command line. Select number 1, so that refers to model 1, and then a slash means chain, and then the letter B. So those are 3 different ways of doing the same thing. And use whichever one is most convenient for you. And for me, which one I use will depend on the circumstances of what I'm trying to do. So now I have chain B selected, and I just want to get rid of it because it's a duplicate of chain A. It doesn't provide any additional information. There's 2 copies of it in the crystal structure. So I'm going to go to the Actions menu, Actions, Atoms, Bonds, Delete. Okay, so now we're left with what you see at the bottom right on slide 72. Okay, so on the Actions menu, if we go to Atoms and Bonds, you'll see for the atom style, there's several different ways of visualizing the structure. And these are called representations. You can show it as a stick, like the ligands are here. We can make it ball and stick. We can make spheres, which are like van der Waals structures or CPK models. And so that's for atoms. We can also show ribbons, like what we've done here. And we can also put surfaces on the structure. We can also color, as we saw earlier, and label from this menu. So, so these various options are very powerful. And remember, they only act on what is selected. Okay, so I'm on slide 74 now in the presentation, if you're following along. So what I'm going to do is I'm going to highlight the dihydrofolic acid ligand, which is in this structure. So remember, this structure is dihydrofolate reductase, and this particular PDB structure has the dihydrofolic acid ligand within it. So let's emphasize it. So I'm going to go up to Select Residues, and here it is. It's DHF, or dihydrofolic acid, or dihydrofolate. And you can see here in green, it's highlighted, but it sticks. So it doesn't really stand out very much. So what I'm going to do, and again, I'm going to show that there's different ways of doing this. I could just, but if I want to make it spheres, I can just click sphere here. And in the presentation, it shows how you can do it from the menu as well. So now I've made it spheres, so each of the atoms is shown as a van der Waals surface or a sphere. And now I want to color it. Oh, one other thing, notice it's still selected. You can still see the green highlights here. So let's color it purple. So now you can see it really stands out in the structure. You can see exactly where that is. So again, it's still we've done several actions. It's still selected. So let's go ahead and clear the selection. So let's go on to the next slide. And now let's view the sequence. So I'm going to go up to the Molecule Display tab and click the sequence icon. And now you can see that the sequence is shown corresponding to this structure down here in a new window. So if we go onto the next slide, which is 76, go ahead and pick some point in the ribbon and control click on it. So I've selected this particular amino acid in the structure, but if you look over to the sequence, you'll see it's also highlighted in the sequence viewer. Now, let's say I want to add additional amino acids to the selection. Well, actually, let me first choose a different amino acid. So if I choose a different one, control click again, the initial selection is removed. So now we have a different amino acid selected. If you want to add to the selection just one more amino acid, control shift click. And now you can see we have 2 amino acids selected here. And as before, you can hit the up arrow button to select more or the down arrow button to select less. So let me clear the selection. Now let me go ahead and select within the sequence. So if I put the mouse in the sequence and do a little bit of a drag move, you can see when I select in the sequence, the corresponding residue on the structure is selected. So the nice thing is that you can go either way. You can select in the structure and see it on the sequence, or you can select in the sequence and see it on the structure. And if you drag a mouse click across several different residues in the sequence, you can see how all of them get selected both places as well. So being able to select in ChimeraX both is very important, and it's really very easy. Okay, so let me go ahead and just select one residue again. And as before, we're going to label this residue just to keep track of it. So I'm going to go to Actions, Label, Residues. Oh, I missed. That's interesting. Huh. Oh, that's right. It's very tiny here. But we can control that by Actions, Label, Set Label Height. And we're going to make it 3. So depending on how big your structure is, you may want to modify this. And also, as you can see from the presentation, there are several different options for setting what the label looks like. And there's also a custom text where you can just type anything as the label. Okay. So next, let's put a surface on this molecule. So I'm going to clear the selection because I want to do it on the entire molecule. And let's go Actions, Surface, Show. And there it is. It's as easy as that to put a surface on the molecule. Now, if I go up to the icons here, I could have just as easily done this from the show icon, but notice there's hide, so I can hide or show the surface. And notice that this particular surface command only does the protein parts. It doesn't do our DHF ligand or the cofactor that was also in this particular structure. If I go to the model panel now, there's a little triangle here next to 4DPD. If I click on that, it's showing me that there are now multiple different parts of this view. There's the structure, there's the label, and there's the surface. And another way I can control the scene is click on and off things using the I column. So I can turn the labels on and off. I can turn the surface on and off. Okay. Now, being able to view surfaces is nice for a lot of reasons. For example, we can, go in here. We can look at how the different molecules bind. This pocket would be the target area that you would do docking with in a drug design campaign. But one other really great property of ChimeraX, capability of ChimeraX, is that we can map properties onto this surface. So if you're in the molecule display tab and you click the electrostatic button, it takes a few seconds because there's a few calculations that need to be done. But what it has now done is it has mapped the local electrostatic charge of the protein onto the surface. So any areas that are yellow have a negative charge associated with them, and any areas that are blue have a positive charge associated with them. So looking at this can be very useful if you're trying to decide how two proteins might interact with each other, how a small molecule might dock into a binding pocket. So there's lots of really valuable information that you can get from this view. So that's the electrostatic coloring. Right next to it on the menu, on the icon bar, is hydrophobic. So what this one does is it maps hydrophobicity onto the surface. So areas that are blue are hydrophilic, and any areas that are brown are hydrophobic. And the way I remember the coloring of this is the blue corresponds to being in water, like, the ocean is blue. And the brown corresponds to being in oil, like, so that's the hydrophobic parts. And so anyway, so these are two very powerful ways of seeing how this structure might interact with other molecules based on its physical properties. Okay, so I'm going to click the hide button to hide the surfaces for now. And now I'm going to scroll back up to the top. And as we saw earlier, there's this let's see. And this sequence right now is much bigger than I really want it to be. So if you go into the separators between the various windows, you get a little tool that allows you to resize them. Okay. So again, as I mentioned earlier, the Uniprot button here is available when you load in the molecule. I'm on slide 80. And if I click on this button, ChimeraX will go out to the Uniprot database and load in some property and information related to the structure from Uniprot. So I'm gonna go ahead and click this now. And it brings up this new dialog box. And this gives us additional ways of selecting various things. Oops. On the structure. And you may have noticed just now, you can dock these external dialog boxes into the actual ChimeraX window, or you can grab them to drag them out. Okay, so show that again. Put it there to put it in, and I'll drag it out. Okay, well, let's leave it in for the moment. So let's look at the first one. I'm going to click on it, and it's showing what is identified as an active site from Uniprot. And you may be wondering why this is far away from the DHF residue that we've been looking at. It's because in Plasmodium, the DHFR protein actually has two functions that are in two different domains. One is DHF reduction. That's the DHFR part at the bottom. And at the top is a thymidylate synthesis, which synthesizes thymidine. So that's what's here at the top, is the active site for the thymidylate synthesis part, where it converts uridine into thymidine. Okay, so that's one of the structural bits that's in that we get from Uniprot. Next is one called the binding site. So this is one near where the cofactor is. This is I think this is an NAD cofactor. Okay, so if we're interested in that, we can use that information. The third one is the domain. So as I mentioned a moment ago, there are two different domains in this protein. There's the DHFR domain, and then there's the TS domain. So if we're just interested in the DHFR domain, we can use this information from Uniprot to quickly select it. And again, here's the entire region around the nucleotide phosphate, the cofactor. Okay. So I'm on, so I've gone through 82, 83, and 84, and 83, and now I'm on 84. So we're gonna go back and click on domain again. And let's simplify the view even further. Since we're really interested only in the DHFR part today. We don't really care about the thymidylate synthase part. So let's not use that. So right now we've got just this part selected. So what I want to do is I want to go to the select menu and use one of the very powerful commands in here, which is to invert the selection. Oh, sorry, I think, yeah, let me do this again. So what this means that everything that was selected before is not selected, and everything that wasn't selected before was. So most of the thymidylate synthase is up here. It's a cartoon. So I can either go to cartoons hide, or I can go to actions cartoon hide to hide that stuff. Okay. And then let's hide all the atoms and bonds. So atoms and bonds hide. So now notice that all of our ligands have been hidden as well, including the ones that we might be interested in, which is the dinucleotide cofactor and the DHF. So what I'm going to do is I'm going to go to the select menu, and normally the select menu does replacements. So if you do a selection, it gets rid of the previous selection and replaces it with a new one. But what I can do is I can change that to add. So now what it does is it adds to the current selection. So I'm going to go to select residues. We know we're interested in DHF. Select residues NAP. And just so that I don't forget, I want to change this back to replace, because I often get into trouble if I forget to do that. And now I can go actions, and now I can show this current selection. Oops, missed that somehow. Let's try this again. So, and if you look at the bottom of the screen, it's telling me how much I have currently selected. So this is usually a good sanity check. So yeah, it looks like I left off one of the commands that we need here. We want to show those atoms. Oh, I forgot to. Sorry, I forgot to change the selection mode. So I'm just going to do this one by one. There we go. Sorry about that. Yeah, if you're on page 85, before it says clear the selection, it should say show the atoms. And as you saw, I did it here just from the show atoms icon at the top. All right. So we have our DHF here. We have a much simplified view of it all, because we've gotten rid of the domain that we're not interested in. We've gotten rid of the second chain at the very earlier. Now let's really zero in on the things that are near DHF. So I'm going to go to select residues DHF and then go to select zone. And this brings a new dialog box where I want to select all of the residues that are less than 5 angstroms from DHF. So if I do that, click OK, now you can see that I'm really zeroed in on the area of interest around this. And for example, let's say I want to see all of the atoms now that are interacting with DHF. Just seeing the ribbon doesn't really give enough information. So what I'm going to do now is show their side chains. So actions, atoms, bonds, show. And you can see much better what is closely interacting with the DHF. And to make it even more obvious, I'm going to color them cyan. So now you can really see clearly what is closely interacting with DHF. Okay. Okay, this is a pretty good place to stop and take another quick break. So again, we'll reconvene at about 25 past the hour. And in the next section, we'll look at mutations, starting on slide number 87. Again, are there any questions?
Yes, Phil, there's one question in the chat box. It's Fuzia is asking, can we predict our study a protein's functionality using ChimeraX, or can we predict, can we predict the impact on the protein's functionality when mutations occur in its sequence? Or does ChimeraX serve only for proteins 3D visualization?
So a protein, ChimeraX is only for a protein 3D visualization. But if there is a Uniprot entry associated with it, like we saw a few moments ago, if you go to the Uniprot page for that protein, there is a lot of functional information there. So you can link, and so you can, from Uniprot, from that particular, and if I find it back here again. So this is the ID number in the Uniprot database. And you can use this to do a search in the Uniprot database to find the information about function. As far as mutations, that's going to be what we're going to look at next. So I'll hold off on answering that for a few moments.
Okay, I have a question.
Yes.
Mamadou Sangare from Mali, African Center of Excellence in Bioinformatics.
Okay, go ahead.
I would like to know if how to access to Uniprot and to show binding site and domain. I lost this part. Okay.
Tell you what, let me use the time in the break to set up for that, and I can start with that.
Okay.
In the next segment.
Okay. Okay.
Okay. Anything else? Okay, thank you. See you in a few moments. Hello, everyone. I'll wait just a minute more for everyone to get back. But to answer that last question, so within ChimeraX, we see here in the log at the top is the Uniprot information. And so the Uniprot code for this particular protein is A7UD81. So if I go to the Uniprot database, and then the search bar at the top, I enter that code, it takes me to the Uniprot page for this particular structure. And if I scroll down, you'll see there's lots of information about the ligands, the various sites, the structures, information about the families and the domains. There are links for this molecule to lots of different other databases, some of these that you're probably familiar with. More information on the sequence. I mean, I'm scrolling, it goes on and on. Similar proteins. Here are where you can set, where you look at structures from either the PDB in Europe, the one in the US, or the one in Japan. Information about the protein family it is. So this is where you can start getting information about the function to see what family it might belong to. So as you can see, there's a lot of information in here. And there's also, as we saw, structural information. And again, we have a 3D live view in Uniprot. This is much more limited than what you can do in ChimeraX, but it is available. And you can pick it turns out there are many different PDB files for this particular protein from different investigators looking at different aspects of it. So, so if we can click whichever one we want, we can load different ones here. Ooh. And if we scroll down, here's 4DPD, which is the one that we're looking at. So if we go back and look in here, this is the exact same structure that we were looking at earlier. And then, as you can see, there are several others. So once you know the Uniprot identifier, that allows you to get a lot of additional information about this protein. So I hope that answers that question.
Okay, yes, thank you.
Okay, so let me. Okay, I think I'm gonna leave, show the presentation here just for a couple of minutes. So, so we're on exercise 4, slide 87. And in ChimeraX, we do mutations based on what are called rotamers. And a rotamer is the different isomers of a molecule that you can convert between by rotation around rotatable single bonds. So usually for proteins, it's the various side chain torsion angles for each specific type of amino acid. Some amino acids, like glycine, where there's only a hydrogen atom, there are no rotamers because there's only one possible structure. But some amino acids, like leucine, have several, and some have quite a few. different possible rotation angles for the various side chain torsion angles. Now, research groups have come up with what are called libraries of rotamers. And essentially what they've done is that they look at existing structures from the PDB and see what is the statistical probability of various combinations of these torsion angles. And then they rank them and give a probability for each possible combination of torsion angles. Now, different groups have come up with different libraries based on different criteria. And ChimeraX provides several, but the one that we're going to use is called Dunbrack, because it was Dunbrack and his research group that came up with this particular one. So the next thing is which mutations to make. So I'm going to go back to the poster that kind of triggered all of this. And here are the 4 mutations that are in the structure that they were interested in. And these are 4 very common mutations for that the parasite makes in this protein to try to relieve activity of various drugs which target this particular enzyme. So, so let's take a look at these various mutations. So I'm going to go back to the ChimeraX window now. So, and I'm going to, first thing I want to clear the selection. And also I want to yeah, I'll leave this here for now. Okay, so the first one we want to select is N51. So there's different ways we can do that. We can start clicking on the structure. Well, there's 102. Here's 40. We can kind of go through it and see if we can figure out where 51 is. Oh, okay, there it is. So that wasn't too hard to find. Or we can go to the sequence viewer. And just like the structure viewer, if we put the mouse cursor on, it will tell us where we are in the sequence. So here's 82. Here's 51. So it was pretty easy and quick to find it on the sequence. Or we can just type in a command at the command line. Select number 1, and a colon is the ChimeraX shorthand for a residue, and then type 51. So all 3 of those ways will work. Okay, well, I've already got the command typed in. I'm just going to use that one now. But there it is. And again, you can see it's selected here in the structure and on the sequence. Now let's look at how we're going to mutate this. So unfortunately, if you go to the menus, there's not going to be a mutate command anywhere. So we go to Tools, Structure Editing, Rotamers. So this is related to the rotamers that we just looked at a few moments ago. And we have one amino acid selected now. It knows that that particular amino acid is asparagine. But in that paper, we want to make this mutation to isoleucine. So if I click on rotamer type and I go to isoleucine, now if I click OK, it shows me all of the different rotamers for isoleucine. And notice that they're ranked in decreasing prevalence. So, and you can see the very first one here has by far the majority of all, is found in by far the majority of all structures that were looked at in the PDB when this library was created. Okay. So on the next slide, I'm going to come back here and click on the first row. And now if we zoom in here, you can see that this first rotamer is shown on the screen. Coming back in, if I use the down arrow, it will now go through this list of all of the different rotamers to see where they go. At the bottom, note that you can also have this tool show any hydrogen bonds or clashes as you go through. So isoleucine, the side chain can't make any hydrogen bonds, so we're not going to be interested in those. But we can calculate clashes here. And in this, oops, where did it go? Sometimes it docks this when you don't want it to. But you can see that it's now showing bumps from the first rotamer here. But as we go through, there are fewer and fewer in some of them. So there are 4 clashes here with the highest ranked rotamer. So that's probably not so good. The next one, there's only 1 clash, and you can see here by the dotted line, this is fairly long distance. It wouldn't take much rearrangement of the overall protein structure to get rid of this. And the rule of thumb is, or the way I like to think of things is, I want to use the highest ranking rotamer that does not have a lot of clashes. So I would probably stick with this one. So, so this is the one that I like, even though it's got pretty low prevalence in many protein structures. So when I have the one I like, I can click Use Chosen Rotamer. And now this is an isoleucine. Okay, so this is how you use the rotamer tool in ChimeraX to make mutations. And once you know that you're looking for the rotamer tool, it's really pretty easy and straightforward. So again, here's the rules of thumb that I usually use. I avoid bumps with the rest of the protein. Sometimes one of the rotamers will look very similar in its shape to the amino acid that you're replacing. So I will often try to pick that one as well. And then as we discussed, you want to use a higher probability rotamer. Okay. So we'll move on now to exercise number 5, which is on slide 92. And this talks about how we can use ChimeraX to interact with more than one model at a time. Now, there are 4 mutations that we're interested in, and we could individually go through and make those mutations one by one. But it turns out that there's another structure in the PDB that already has all 4 of those mutations. Actually, there's a few structures that have all 4 of those. But one of them has the drug pyrimethamine already in it, in the structure. And pyrimethamine is one of the things that was being looked at in the study from the abstract that started all of this. So that drug has a PDB code 3QG2. So let's just go ahead and open that. So I type open 3QG2. And I'm going to scroll back out. And as before, you can see there's a lot more to it than the part we're looking at now. We get all of this information. We get the same Unipro code because it's the same protein. We'll notice here that there's different small molecules. This time, instead of DHF, it has one called CP6, which is the drug pyrimethamine. And if we scroll in on the part of the DHFR that we've been looking at, you'll see that there's a reasonably good overlap between the two when I read them in, but this is not very typical. Usually the two structures will not overlap very well when you first read them in, even if there are similar molecules. Okay, so let's go forward to slide 93. So as before from the log, you can see that there's two chains, and we're going to simplify just like before. We're going to click chain B, see that it's highlighted, or you could do it from the command line, and then we're going to delete it. So actions, atoms, bonds, delete. Let's get rid of those. Okay. Okay, so we know that there's a drug that we're really interested in this particular structure. And if we go to the log here, when we read it in, if I click on the label CP6, it should select it in the structure. It doesn't look like it did. Oh yeah, it did. It's just hidden by the DHF from the other molecule. So actually, I'm going to go ahead now to the model panel and turn off the one that we were looking at originally by clicking in the I column. So here's the DPD that we got from clicking here in the log. And we're going to do just as before. We're going to show it as spheres and color it in red. So there's the DPD. And now I'm going to clear the selection by control clicking in the backdrop. If we go back here in the log, oh, I guess this doesn't work in Windows either. Okay. Sorry, one moment, please. Oh, that's right, I forgot. So I clicked on the letter CP6, which is the name of the ligand. One thing I forgot to do is if you click, I mean, it's shorthand name. If you click on its full name, it brings up a browser window, which shows its two-dimensional structure as well. So if you want to see what this ligand looks like in a more simplified view, you can get it here. Let's go on now to slide 97. And I'm going to make the model panel bigger here. And you'll see that there are now 2 entries. There's the original one that we had from 4DPD, and there's a new one from 3QG2. And we can interact with these independently, as we just saw a moment ago. So we can turn one off, we can turn both of them off, so we can interact with them however we want. So if we go on to the next slide, number 98, you'll notice that, like I mentioned earlier, they're fairly closely aligned, but they actually could be aligned better. So ChimeraX has a tool which allows us to align protein structures to each other. Now, what it's doing, it's aligning the structure based on the sequence. But the most important thing in this alignment is the structure. It's not the sequence. So if we go up to the Tools menu, Structure Analysis, Matchmaker, it brings up this box. And by default, and there are 2 columns here. One has the reference structure. This is the structure that is not going to move. And then the structure or structures to match is on the right column. So this is the one that gets moved to align onto the reference structure. Now for right now, we can leave all the default parameters. And right now it's best aligning pair of chains between the reference and the match structures. Just quickly show though that you can also align specific chains in the reference structure with the best aligning in the match, or you can go to specific chains in each structure. But I'm going to go back to the default, which is just let ChimeraX do all the combinations and find which chains are most similar to each other and do the alignment. Okay, so I'm going to click OK. And you can see here, the alignment between the 2 is now much closer. In fact, you can see this ribbon here, it shows colors from both of them as you move it around. So this alignment is very close. So the point being here that ChimeraX is has a very powerful tool for aligning proteins based on their structural similarities. All right, so so now we have the 2 structures aligned, and what I haven't shown you yet is how you can save your work. So ChimeraX has what's called a session file, which will save all of the information that is currently in this session so that you can recall it later. So if you go up to the File menu and you go to Save, and Files of Type, make sure that ChimeraX session is shown. And you'll notice that the file name extension is .cxs here. And then what you can do is you can navigate within your file system to wherever you want to save it and just type in a file name, like, you know, workshop. And if you click Save, you'll now have this current progress saved that you can recover later. All right. So let's move on to slide 101, which is exercise 6, which is how ChimeraX uses AlphaFold. Now, something I haven't mentioned before, if you look down here, you'll notice that there is a dotted line here. And for now, I'm gonna turn off the newer structure. And remember, the newer structure already has all 4 of the mutations in it. So we can use that for doing various kinds of comparisons. And don't really have time to do much of that today. But what you would do is identify where those mutations are in the active site, relative to the active site, and see if they were likely to affect the binding of the DHF ligand or any of the drugs. Now, the dotted line corresponds to a sequence of amino acids, which was in the solution where the crystallographer made the crystal, but they are not visible in the crystal structure itself. And what this means is that these are disordered in the crystal structure. So there's no visibility of them. So in ChimeraX, those residues are connected by a dotted line. One thing of note is if we turn on the structure with the 4 mutations, those residues actually are visible in that structure. And sometimes you may want to use Matchmaker to align various structures and see which parts see if there are any parts that are missing in one that are visible in another, and get additional information that way. If we go over here to this part, you can see here's some missing residues that are missing in the newer structure. But there is an additional way that we can look at all of the amino acids in one structure, and that is to use the AlphaFold database. Because AlphaFold will give a structure prediction for everything, and then it'll give you a confidence score to go along with it. And ChimeraX gives a real simple tool for doing that. So if we go to Tools, Structure Analysis, oh no. Oh, this must not be the right version of ChimeraX. Shoot. Hang on one moment. I'm going to quit out of here. Okay, this should be it. Oh, I don't, This one should have it. I must have gotten a daily build where it didn't build properly. I'm very sorry about that. Hopefully, yeah, so this is one of the downsides of getting the daily builds, because they will have all of the newest features, but occasionally the ChimeraX development group breaks things when they're trying to add new features, and some things that should work don't. So that's probably what happened here. I hope none of you are having the same issue. So for this part, I'm just going to have to go ahead with the presentation part. So if you choose this menu item, and make sure that the sequence is set to #1, which means that it's the original DHFR molecule that we were looking at. If you click the word fetch, what it will do is it will go to the AlphaFold database and load that structure into your ChimeraX session. And that structure should look something like this if you do it. And again, I've recreated the color coding here. Most of it has high confidence in the darker blue, but you will see that there's a couple of loops here, and that there are no dotted lines because the entire structure is shown. And again, both the DHFR and the TS domains are dark blue, they're well represented, and you can have high confidence in the structure. And in fact, you can use Matchmaker, Chimera automatically uses Matchmaker to do the alignment for you. So you can see in your session, if you've done it, how that works. And that would, so if you turn on the two visibility checkboxes in the model panel, you'll be able to see how closely the alignments are between them. And you can see, again, no dotted lines. You can see where the AlphaFold has filled in the structures. Now I'm going to go back a couple of steps to the AlphaFold dialog box. You'll notice that there's a couple of other important buttons on here. So fetch is the one that finds from the database the structure which is the most similar to the sequence. Search, on the other hand, does a blast search of the AlphaFold database and will show several related structures, and you can pick whichever one you want to do to load into ChimeraX. As you can imagine, that's going to be a little bit slower. And then slowest of all is if you have a brand new sequence which might not have any related structures in the AlphaFold database. For instance, if it's from a species which is not in the AlphaFold database, one of the 21 species, you can use the predict button, and it will actually use the AlphaFold II program to calculate a new model for that sequence. Now, this can take a while. This can take several hours to do, and you have to leave your ChimeraX session running the whole time. But it works, and it will often give you a high quality structure for something brand new that no one has ever looked at before. Okay. So now we'll go on to exercise number 7, where we're going to examine some molecular docking results. And as I mentioned here, we're going to start fresh. So, and it didn't matter that I wasn't able to show you on the screen the AlphaFold results, and hopefully some of you got to see those. But anyway, I'm going to close the session and then open for DPD again. All right, so we have it open again. We are going to simplify the view one more time. I'm going to go through this more quickly this time. So I'm going to select chain B in the log, and then from the actions menu, atoms, bonds, delete. I'm going to click the Uniprot in the log. And I'm going to click domain in the dialog box here. And then I'll close this. From the select menu, invert. And now I'm going to do it a little bit differently this time to really set up exactly what I want. I'm going to change the menu mode. So select menu mode, I'm going to change it to subtract. Because right now, if I delete or hide, I'm going to delete some of the ligands that I'm interested in. And in this case, it's the NAP cofactor. And I want to keep that when I do show the docking results. So I've got select minus. And now if I go to the residues, I'm going to select NAP. So that's no longer selected. That's right in here. And then, and now I'm going to delete everything that's selected. So select atoms, bonds, delete. And now I just have my DHFR domain with the nucleotide cofactor in it. And before I forget, and I left this off the directions, I want to go back to menu mode, replace. So I'm in my normal menu mode because again, yeah, the menu, the select menu won't work the way you expect if it's set up wrong. Okay. Now, in the same online folder where you were able to download, the presentation, there's also one data file in a separate folder there, and that data file has the name DHFR ligands.mol2. So again, this is one of those file formats that contains molecules that we talked about earlier. This file contains a set of ligands which were docked into the DHFR protein. Okay, now full disclosure. This really isn't from a docking experiment. These are actually ligands from different crystal structures where the DHFR proteins were aligned. But the idea is the exact same because I couldn't find any docking data for this particular protein. But the ideas are exactly the same. So if you use a docking program like say Autodock Vina or Maestro or various other docking programs, ChimeraX has a way of loading the docking results of the small molecules in and analyzing them. So what I'm going to do first is I'm going to go to File, Open, and it's on my desktop. So there's DHFR ligands.mol2. I'm going to click Open, and here they are. So all of these molecules have been docked into the DHFR binding site. And you'll notice in the model panel, they have an ID of 2. And if you click the little triangle to open it, you'll see each one is in a different submodel of number 2. So the first one is in 2.1, 2.2, 2.3, etc. And right now they are all visible. So let's now analyze this data, or these data. So if we go to the Tools menu, General, the bottom is this command View docx. And it puts this new panel in here, and I'm going to pull it out for now if I can. Oh, it didn't like that. And you can see here, essentially it has a table of the various results. So the name column has a shorthand description. The description column has a longer description. This is the name that's actually going to be in the ChimeraX session. So if we go to select residues, you'll see this is the various names, the shorthand names for all of those small molecules. Here's its ID in the ChimeraX session. And here's a number score, just the way they are loaded in from the file. And there's also an energy score here. This is inside the file, and it's a measure of the docking score that the various docked ligands have. And remember, for energy scores, lower energies are better. So if we can look at this, this one here, minus 49.12 is the best one. And this one here, number 7, negative 38.87 is the worst one. So there's all of these different molecules in the table. I'll leave that there for now. So I'm on the slide 107 now. And you'll notice that there's some additional buttons here at the top of the table. Graph, you can make graphs and plots, add hydrogen bonds and add clashes. And you can also save these results. But anyway, I'm going to first do a plot. So I click a plot button and it brings up some controls over here. And I want to set the x-axis to number. So it's showing the various category, or the various columns in the table. And I'm going to set the y-axis to energy score. So now we can see a plot of the energy score for all of the various ligands that are in this docking file. All right. And let's click on the dot here for number 4, which has the lowest energy. And you'll see that that one is now the one that is shown in the table. So we can see exactly how that's docked in here. Okay. So I'm going to go on to the next slide now, which is 108. And now here's the table here at the top, it docked in for me. So I'm going to click on this row containing pyrimethamine, which is the drug molecule. And now it's showing the drug molecule here in the binding site. And once I have that row clicked in the table, I can use the up and down arrows on my keyboard. And you'll see in the table above, as I'm using the arrows, I'm scrolling through the table and I'm scrolling through viewing all these different docked ligands in the active site. And let's go ahead and select row 3 here. As you see from above, row 3 contains DHF, which was the original native ligand, DHF for DHFR. But as we go through this, look how many different kinds of binding modes or binding poses there are among the ligands in this docking study. So this is a really great way to look at some of these. And in the abstract, the poster abstract, pyrimethamine and cycloguanyl are the 2 drugs that were being looked at related to this genetic study of these four mutations. Okay, so let's go on to the next slide, which is 109, and go ahead to the top of the table and click Add H bonds. And now, if pick one again, and now you'll see that are little blue dotted lines showing where there are H bonds between the various ligands and the protein structure. And you can see some of the different ligands have different H bonds. Some ligands don't have any at all or very few, like this one. There might be one back there. No, this one doesn't have any. And so this is another way to help you gauge which ligands are really, besides the energy, from a structural point of view, this helps you identify which ligands are really binding well. And if you're working with a chemist can look at some of these and notice where there are modifications they can make to these molecules, where they might bind better or add additional hydrogen bonding capability to the ligand. You might be able to do that by synthesizing some new molecule which combines multiple ligands, which has hydrogen bonds in different areas. All right, go on to the next slide, which is 110. And if we scroll up to the top of the ViewDocX table, now we're going to click the Add Clashes button, which shows places where there are bumps between the structures. And let's see here. So, and these are shown as purple cylinders. So you can see, here's some. And again, as I go through, there are different purple cylinders indicating bumps for the different structures. And we can, again, we're only showing it a little bit here. Let's see if I can make this any smaller. No, it's not. Oh, here we go. So we can see more. Notice that there are new columns in the table also showing the numbers of H bonds that can help us guide what we're doing and of clashes. And we'll see that this particular structure, BT3, has 8 clashes. So select that one now, and we'll make the graphics window bigger again. And you can see how there's, clashes all over the place. And if you look at the energy scores, this will be the one that has the worst energy of all of them, because it has all of these clashes. So there was a doc pose, but yeah, it really wasn't very good with all of these clashes. Okay. So one final thing is let's get a real visual view of how these die in the dining pocket. So I'm going to go up to the model panel, and in the 4DPD row, I'm going to click this row, and to select it, and then I'm going to go up to the I'm going to close this out now, the ViewDoc-X panel. And under the model display tab, I'm going to show the surface again. And now you can look exactly how the different ligands are binding here into the surfaces. You can see that in this particular binding pocket, there's a small opening in the back and a big opening in the front. And we can do all kinds of fun stuff with the various ligands, looking at how they fill this binding pocket. Now we can do this reasonably well on a computer screen, but this is really much more powerful way of looking at this is in virtual reality. So we're going to take another quick break here and again come back at about 25 after. And so we're going to look at in virtual reality, look at the these docked ligands here in this binding site. I want to point out now that ChimeraX has this virtual reality capability. It can also share the same meeting with multiple users. And those users can be either across the room or across continents. And in fact, we've had meetings, shared meetings between different people here in the United States and different people, different users in our visualization lab in Kampala. And sharing the same views, the same interactions with the structures. So after we look at the DHFR binding site, we're going to take a little tour through the SARS-CoV-2 virion and its spike protein, and then look at some medical images. So let's come back in about 10 minutes and we'll continue on with the VR. Any questions?
No.
Okay, thank you. We'll be back in about 10 minutes. Hello, everyone. Let's get started. So we're going to go into virtual reality now. And I think I'm sharing my camera. So this is a virtual reality headset. And I am going to put on this headset, and with these controllers, while I'm in virtual reality, I'll be able to control the ChimeraX scene. And this gives a totally different and really unprecedented way of looking at these structures. So let me go ahead and go into VR, and I'm just going to stop sharing my camera here, and let me go into VR. So can you see what I'm seeing here in VR on the screen?
Yeah, we can see.
So this is the same molecule we've been looking at, but to me, and these are what my controllers look like in the VR scene. This looks to me like it's about 3 or 4 feet tall. And you can see here's the binding pocket with one of the ligands in it. In the back end is the dinucleotide cofactor. And the reason I left this in was because it interacts with the docked ligands, and you can see that much more clearly here. So, so far, it probably on the screen looks pretty much the same as what you see so far. But what I can do is I can make this much bigger. And now I can walk into the pocket and look around. I can look up and back where I came from. And now this binding pocket looks like it's about 30 feet tall to me. Okay, so I can now see a much clearer look at how the shape of this ligand fits into the binding pocket and how it interacts with the other molecules. I can see back there where the hydrogen bonds are that it's making with the protein. But I'm not just held within the pocket, because I can go over here and I can go through the surface. And now I'm just looking at what the inside of the protein looks like here in this location. So I can see which side chain has the hydrogen bond through this surface with this ligand. And again, I can also see where it came from, where the big openings are, how the ligand can come into the pocket. It's just an unbelievably powerful way of seeing how different ligands bind in. So we have this ligand in right now. I'm going to change over to a different one. Now we have a different ligand from our docking. And you can see this one fits in quite a bit differently. And it also has some bumps. You can see this one has a bump here with a cofactor. And so this is just an unbelievably great way of interacting with docking data. So let me stop here and get a new ChimeraX session going. So I'm going to stop this one. And then I'm going to open a new one. So we're going to take a look at some aspects of the SARS-CoV-2 structure. And let me first here just finish loading in one more bit of it here. Still loading, just one moment. And you can see that this is also in the full coronavirus virion is also in the scene. And I'm going to start by turning off some parts of it. All right. And even more importantly, I'm going to share a common meeting with Megan. So she's across town in another headset. And what I'm going to do is start up what's called a ChimeraX meeting. So if we go to Tools, General Meeting, I think I missed it. And I'm going to give the meeting the name Workshop. And Megan, are you on? Are you available? Hello, Megan? I can't hear you. Megan, I can't hear you. Can you hear me? Can someone tell me in chat if you can hear me?
We can hear you.
Okay.
Paul. I can't hear Megan.
Okay, thank you. Megan in chat says she's ready.
And she's also unmuted, but like, I think there's issues with the sound from their part.
She may, yeah, it may be a sound issue from her end. Well, let me start the meeting. So Megan, if you can hear me, I'm going to try using the ChimeraX standard meeting to start with. Oh, there she is.
Yeah, sorry, I was trying to switch microphones. I'll just leave it from the computer.
Yeah, that's what I did. I think it's easier. You were able to hear me, right? So
Yeah.
Okay. So I'm going to choose just the regular ChimeraX meeting server. I gave it the name Workshop, and I'm going to create a new meeting. And let's see if this works. Okay, so my meeting has started. And now Megan should be able to join this meeting that I've just created. But I'm going to go back into the VR headset now. And let me start my video so you can see me moving around.
And I'm joining, it's just pulling down the data.
Right. Okay, it lost its connection here. Oh, well, the trouble with live demos.
Bill, I don't know if you've got VR on.
Yeah, it's definitely on.
Oh, okay, all right. I wasn't seeing it on your screen. So
Yeah, no, the session has, I'm going to go VR off and then VR on again.
okay.
So you will, but it shouldn't affect your session. So. Well, that looks better. Okay. Wow. That's not better. Okay, here we go. So can you see the session? No, I see you haven't joined it yet, Megan.
Yeah, my computer is just thinking.
The problem is that I added the full virion. So I'm going to stop. I'm going to get rid of this from the session. So, but anyway, before I go on, this is a model of the full SARS-CoV-2 virion. And if you look very carefully here, some of the spikes, the red ones, have one part that's sticking up higher than the other, than the lighter colored ones. This is with the receptor binding domain, and it's constantly flipping up and down between those two positions. And it can only bind to the receptor when it's in the up position. Okay, Megan, I'm going to go ahead and delete that from the session and start over.
Okay, I'm just restarting ChimeraX.
Yeah, so let me go ahead and just start all over.
All right.
Okay, so okay, so the meeting started, and again, the name is workshop with a capital W.
All right. I had some issues earlier, so I'm just trying again. After restarting, let's see. Sorry about that.
So I'm going to go ahead and then just join when you can, because it will be obvious when you join. So, so you can move the objects with these controllers, and if it's a long way away, you can use kind of like a swimming motion to pull it towards you. And notice that you can also put additional 2D images in the ChimeraX along with you. So if you're doing training, and this training could be across far distances, we got it working right here today. And so this is just some various features of the coronavirus. So here's a electron microgram of some coronavirus virions, and you can see kind of blurry the spike proteins that are sticking out from the outside surface of them. And then here is a picture of the model that we were looking at a few minutes ago. And if you look very closely again here, you can see that some of them are up and down. And then let's go take a look at our spike protein here. Right now, the spike protein consists of 3 what are called protomers. One of them is blue, one of them is red, and the third one is shown just the ribbon structure. So you can see the internal structural detail. The blue and the red ones are shown as surfaces. And the ribbon one is colored by what's called a rainbow coloring, which uses the spectrum from blue at the N terminal to red at the C terminal. And you can see down here, there's one amino acid that's got a different color. This is indicating the position of the start of the transmembrane domain. That's the outer surface of the outer surface of the viral membrane. And in fact, so that the membrane goes from about these 2 positions here, and then the spike is sticking out from it. So this bottom part is actually inside the viral particle itself. So we can see here there's the 3 different structures. If we go up to the top and we kind of really zoom in here, you can see that there are some parts here that, that are a different color. Oh, sorry, I bumped my head with a controller. Some parts of it have this magenta color. And these are the key amino acids which were mutated to proline. So there's 2 adjacent amino acids which are mutated to proline. And when you do that, it stabilizes this spike structure. And that's the basis for all of the vaccines that are created that are using the spike, that they're based on mutating these prolines together. I mean, these 2 residues to proline, and that locks the spike into this particular structure. Otherwise, it can kind of fall apart. So that keeps it together for the purposes of the vaccine. And as before, we can really blow this up really big. Even go inside of it. You can see all the interior structure here. Let me go find one of these alpha helices over here. In VR, it's really cool to be able to look down, an alpha helix and just walk through the alpha helix as it spirals around you. But you can get a lot of insight into the various structures, how they fit together in VR, because it's so immersive and around you. Let's see,
Oh, you can keep going. I'm having some technical issues, but I hopefully will get this sorted shortly.
So even if you are in VR, you can turn on your menus here. And so for instance, what I just, and I'm gonna, actually something happened earlier. I'm going to, I'm going to restart again as well. I think that'll give you a better chance to join.
Okay, I've got it running now.
Oh, you do? Okay.
Yeah. Will I go ahead and join?
Yeah, go ahead and join them.
All right.
One other thing I forgot to mention is that you can move the various objects independently of each other. So, for example, we talked about the right mouse button menu earlier, so I'm going to click on that now. And now I can assign any of these controls to any of the buttons on the controller that you can see. So for example, I am going to click one of the buttons on this controller to be drag model. And now I can point the controller at one of the objects, for instance, one of these two-dimensional figures, or the spike protein itself. Up here is an antibody, and I can actually, I know that it docks right about here somewhere. And this is up here, this is the part that I talked about earlier, that this is the part that flips up and down. So only in the red protomer is it up. Normally, as in the other two, like the blue one here and the ribbon one, it would be down. So it would be in, it's going to walk around it over on this side. But when it's up, this particular antibody can bind to it. Also, and things got moved a little bit, but also this particular, this is the ACE2 receptor, and it also binds into this region here. And it is only can bind to it when this part of the spike protein, which is called the receptor binding domain, flips up. It flips up and the receptor can bind to it. But if there's an antibody here, then this can't bind to it. This can't bind to it if there's an antibody here. Now, one of the great things about the vaccine is it doesn't just generate antibodies here to the receptor binding domain. It also generates additional antibodies that bind over here to the N-terminal domain. And you can see what I'm doing. I could align these structures so that they are already aligned. But what I'm really kind of doing right now is doing some manual docking, seeing where the two structures can fit together. And this is something that you really can't do on a computer monitor. You really need to be in virtual reality to do this sort of thing.
Hey, Phil, I'm getting a message that it's waiting for scene data from meeting host. So.
Okay, so let me try putting it out and coming back again then, because it would be really great to be able to show this.
I know, yeah, it's awesome.
Show the meeting capabilities.
And again, for everyone, you can do this without VR, which, you know, if you're in a Zoom meeting like this, as we watch it all along, Phil is able to share his screen so you can see what he's doing. But with the meeting command, you can actually let other users control the scene, whether or not you're in VR.
Okay. Yes, I'm just going to go back to the beginning.
Okay, great.
Okay, so yeah, so the meeting has started now, and I started up Steam again as well, Megan. So
Great.
Okay,
Okay, I'm receiving data now.
okay, great.
And this will take a minute because it's a big file. But the nice thing about the VR is it will work regardless of your bandwidth. So once you download the scene, you have a local copy of the file. So the information that's being transferred doesn't necessarily require a fast data transfer. You're only sort of passing the coordinates of the other person. Bill, is that correct?
Yeah, that's right. So is it still loading for you, Megan?
It says received scene data. All right, so do I need to start SteamVR?
I think all you have to do is VR on. Oh, and I don't know if you can see the grid that sometimes appears or not, but this is sort of like the extent of my play area. So if I go out of bounds, if I cross over this boundary and I'm no longer in the area that the VR recognizes. So if you've been seeing a grid appear and disappear, that's the reason for that. Any luck, Megan?
Now I'm just having a SteamVR issue, but just because it's a live demo.
Because it's a live demo, of course. Well, in case it doesn't work, what I would see when Megan joins the meeting is I would see wherever she's standing in the scene, a picture which shows where she's facing. And I will also see two different colored controllers that show where her controllers are. And each one of us has control of the object. So I can move things around now, or she can move things around if she wants. And we can move so that we're both standing in the same position and looking at things from the same angle. We can see where exactly each other is pointing in this three-dimensional space. And I know it doesn't really convey on the screen, but right now it looks like this spike protein, to me, it looks like it's about 50 feet tall, 50. So, and when you get into a structure that's this big and you can walk around in it, you just get a completely different concept of the three-dimensional arrangement of all the various components in it. So I think we're running out of time. I think let's move on.
All right, wait, I'm here, sorry.
Oh,
We got it.
Oh, is it still loading?
No, I'm good, I'm in VR. Can you see me?
Oh,
There you are.
there's Megan.
Hey.
Yeah, so sorry about all of the, I'm glad we got this to work because it's just really powerful. So Megan can see where I am. I can see her. I can see her controllers. She can move the models around just like I could. She can rescale everything.
So this is great because now I can go inside, and if say Phil has his pointer, so I can sort of, hold on,
I'm gonna come to about where you are.
there we are. So I'm right here next to Phil.
Now.
And if Phil wants to point something out to me, I can see right where he is, and we can talk about this.
Here's the mutations, and there's a whole series of commands available from that right mouse button menu. Like I can do things like label individual residues. Actually, let me show one of the really great ones here.
And Phil, you can hear me okay, yeah?
Yeah, I can hear you.
Oh, great, perfect.
So here's the tape measure button. So I clicked on this to assign it to this button here. And you can see that the icon for this particular button now matches the icon on the menu. And so if I want to find out how far it is between, say, this atom and this atom on adjacent protomers, I can just drag out and measure exactly three-dimensional space and see what it is. And this kind of thing, three-dimensionally, is just really difficult to do any other way. And if I go back out here again, you can see there's all kinds of things that I can do. Like, for instance, I can drag individual models like I showed earlier. Like if I go out here, and that's how I was able to move one molecule relative to the other ones. Again, very powerful. You can do labeling. You can even do mutations using the rotamers within here. So anyway, it's just extremely powerful. So, so Megan, I'm going to go ahead and go on to the medical imaging now.
Great.
So I'm going to go VR, turn off VR. Close this session. So this is a micro CT scan of a mouse femur that was used in a drug study to see how it was affecting bone density. And as you can see here, oh, hang on a second. As you can see here, just like a molecular model, you can load it into VR. You can show it as a surface or as a volume. So you can kind of see more of an X-ray view of it. You can see some inside structure here, and we can do it either way. I'm going to go back to surface for just a second. And now let's go into VR again. Where are you at? Now remember, this is a mouse femur, but I can make it as big as I want. I can view it from all angles, and I can even go inside, and you can see all of the interior connecting structures inside of this femur. And it's just, you know, I'm walking around in it. And you can also see that this even structure in this outer surface of the bone here, if you blow it up. And I'm kind of in between the inner and outer parts of the bone here where the marrow might be, as well as on the inside of the femur. So anyway, so this is one example. We also have I mean, you can look at any kind of CT image, whether it's a brain scan or viewing a lung. So just wanted to quickly give you a feel for some of the power that's available within ChimeraX and with VR. And Megan, if you want to take a couple of minutes to show off pathogen AR.
Yeah, let me see. I'm gonna I'll close out.
I'm going to stop sharing.
And let's see my
So while we're waiting for Megan, are there any final questions?
All right, so I'm going to see if I can get this on my webcam, all right?
Okay.
Okay, so we're doing this kind of like with, and I've got my office in the background here, but you can see this is my iPad. You can do this on a smartphone, whether it's iOS or Android. And this is that model of SARS-CoV-2 that Phil has shared that we looked in VR. We saw a 3D print of it earlier, but now I can take this with me. I can zoom it, I can zoom in and out, I can turn it around. And so then Pathogen AR brings me on to see what our next scene is. Okay. And this is actually a really cool feature with the spike protein. If you can see, and again, we're doing this demonstration and you've got my office in the background, but there's an animation here of being able to open and close and see the different configurations. And you guys can download this for free and go and play around with it yourself. There's a couple different modules there. So I encourage you to go and check it out.
The link to it is on slide 115.
So it's a pretty cool tool. And we think it's nice to make some of these models more portable for, oh gosh, we've got wild background there. There's a 3D printer over there and all kinds of stuff. We thought this is a nice tool to make these models accessible wherever you go and to anyone who has a smartphone or a tablet. So we hope you'll check it out. Pathogen AR, you can find it in the iOS or the Android stores. And I have VR headset here right now as I normally do. That's a work hazard.
Yeah, so here's slide 115 and oops. And down here is the link, short link to it. And essentially you can see the same sorts of scenes that we were looking at in VR of SARS-CoV-2, but in your own room, in your own hands. And so that's the end of the workshop. I hope it was useful. And there's a contact information for us on one of the first slides. So please don't hesitate to contact us if you have any questions. And one thing I want to say about the virtual reality is it's not extremely expensive. The headsets are only, I mean, they're not, they're a few $100s but to take full advantage of them with ChimeraX, you also need a PC with a graphics card in it. But other than that, the actual hardware is fairly accessible for organizations to be able to get. So you might be able to convince somebody where you're at to look into it. And we'd be happy to help give guidance for that.
Yeah, please reach out to us if you have any questions on any of this. And if you do have access to some VR hardware, we would love to share that with you and get in a remote meeting session.
All right, so I think that does it for us. We went a couple of minutes over. Sorry about that. And sorry about some of the technical issues that we had. If there's one final chance for questions, and otherwise, we'll look forward to hearing from any of you if you have questions or comments.
I'll just take this opportunity to say thank you very much, Phil and Megan. It was very interesting and it was long, but I can see the fellows or the trainees really stayed on. So I'm sure they will be able to implement this in their work.
Great, great. Well, thank you all.
I see Megan has posted the email addresses. Thanks so much, Megan.
Thank you, everyone.
Bye-bye.
Thank you. Bye.
Thanks, everyone. Bye-bye.